Oh, hi everybody. Today we are going to talk about how cells organize and store genetic information. Specifically, we'll define what genes are, distinguish between genotype from phenotype, and compare prokaryotic and eukaryotic chromosomes, discuss and discuss why extrachromosomal DNA is important. So even though DNA is microscopic, cells have to organize an enormous amount of information in a very small space. The way they do this is critical for survival. And by the end of this module, you should be able to define gene and genotype and differentiate genotype from phenotype, describe chromosome structure and packaging, compare prokaryotic and eukaryotic chromosomes, and explain why extrachromosomal DNA is important in a cell. Alrighty, so let's start out simple. The most important concept in biology, maybe, is the idea of the gene. A gene is a segment of DNA that contains instructions for making some functional product. Usually that product is a protein, but sometimes it is a functional RNA molecule, right? We talked a little bit about those in the previous lecture when we talked about tRNAs and rRNAs. Genes are the unit of heredity. They influence traits by directing cells to produce molecules that carry out cellular functions. For example, a bacterial gene might encode an enzyme that breaks down lactose, or it might encode resistance to an antibiotic. In humans, genes can influence traits like eye color, blood type, and then sometimes with multiple genes, you have things like how tall you are, um, or even like what color your hair is, so forth and so on. As you look at this image, though, notice that a gene is only one specific region of the much larger DNA molecule, which is just packaged together into what we call a chromosome. Now, we need to distinguish between genotypes and phenotypes. Genotype is the genetic makeup of an organism, the specific alleles or gene versions that it possesses. The phenotype, on the other hand, is the observable characteristics produced from that genotype interacting with the environment. So, genotypes refer to the genetic information itself, while phenotypes refer to what we actually observe. For example, two organisms may have different genotypes for flower color, resulting in different phenotypes such as purple or white flowers. In bacteria, a phenotype might be whether the organism survives exposure to an antibiotic. It's important to remember that phenotype is not controlled only by genes, though. Environmental conditions also influence how genes are expressed. So you can have a genotype that allows for the expression of a specific gene, but that gene may only be expressed under certain circumstances or maybe not at all. Now, the chromosome. A chromosome is a structure that contains DNA and associated proteins. One major challenge for cells is that DNA molecules are extremely long compared to the size of a cell. So cells must package DNA in a compact and organized way. Most prokaryotes have one circular chromosome located in a region called the nucleoid. Now, in this image, we have our cross section of a microbial cell. Notice there is no nucleus. There's no membrane-bound nucleus, but the genetic material is kind of squished together a little bit in the center of the cell. It's a little hard to see in this, but this is also circular in nature. We can see that, or we can kind of see that the DNA like loops back in on itself, which is different from eukaryotic chromosomes. Here's a better image of the circular DNA molecule. So even though prokaryotes are relatively simple cells, they still must compact their DNA. They do this using several mechanisms. One is supercoiling, where their DNA twists upon itself. They also use DNA binding proteins that help organize and stabilize the chromosome. Now, finally, the chromosome forms loops, looped domains that further compact the DNA. Together, these strategies allow a large DNA molecule to fit inside a very itty bitty tiny bacterial cell. Eukaryotic chromosomes are much more complex. Unlike prokaryotes, eukaryotes usually have multiple linear chromosomes. These chromosomes are located inside a membrane-bound nucleus, which we can see on the screen here. Eukaryotic DNA is wrapped around proteins called histones This wrapping forms structures called nucleosomes, sometimes described as beads on a string. The DNA then coils and folds into increasingly compact levels of organization. This packaging is important because eukaryotic genomes are extremely large, like bigger than your prokaryotic genomes. For example, if stretched out, the DNA from one human cell would be about 2 meters long. So a single cell. Your cells are microscopic. They contain DNA that would be up to 2 meters long. Now, not all DNA is part of the chromosome or the genome. Many cells, especially bacteria, contain extra chromosomal DNA called plasmids. Plasmids are usually small circular DNA molecules that are separated from the main chromosome. These molecules often carry genes that provide an advantage under certain conditions. For example, plasmids commonly carry antibiotic resistance genes. They may contain genes involved in toxin production or specialized metabolism. One reason plasmids are so important is that they can move between bacterial cells through horizontal gene transfer. This allows useful traits such as antibiotic resistance to spread rapidly through the population. Scientists also use these plasmids extensively in biotechnology for cloning genes and producing proteins. So, in summary, genes are segments of DNA that contain instructions for functional products. Genotype refers to genetic makeup, while phenotype, pardon me, refers to observable traits. DNA must be highly organized into chromosomes. Prokaryotes generally have one circular chromosome, while eukaryotes have multiple linear chromosomes packaged with histones. And extrachromosomal DNA, such as plasmids, can provide cells with important adaptive traits. So with that, thank you for listening to this lecture, and I'll see you at the next one.